Fertilization is the process of fusion of male gametes with the female gametes during sexual reproduction. In the angiosperms, that is, the flowering plants, the male gametes are produced in the stamens and the female gametes in the ovary of the flower. The male gametes are transported from one flower to the other or from one plant to the other through pollination for the purpose of fertilization. Abiotic and biotic agents help in the transport of male gametes in pollination. The pollen grains containing the male gametes land on the stigma of the ovary after pollination. They germinate on the stigma and form a pollen tube that grows down the style of the ovary carrying the male gametes. Inside the ovary, the female gametes are present in the embryo sac. The pollen tube releases the male gametes inside the embryo sac. One of the male gamete fuses with the egg cell to form the zygote. The other male gamete fuses with two polar nuclei to form the endosperm. Zygote is the diploid cell formed by the fusion of two haploid gametes and develops into an embryo. The endosperm is a triploid cell formed after the fusion of three haploid nuclei, and it serves as the nutritive tissue for the embryo. The fertilization in angiosperm is often referred to as double fertilization, as it involves two separate fusion of gametes. After fertilization, the ovule containing the embryo develops into a seed and the ovary forms the fruit. The seed will give rise to the next generation of plants after emerging out of the fruit. Fertilization or fusion of gametes is thus the first step in the continuation of generation in plants by sexual reproduction. Let us see in detail about the process of fertilization. The stamen is the male part of an angiosperm flower. The male gametophyte or the microgametophyte develops within the microsporangia or the pollen sacs of the anthers on the stamen. The pollen grains are produced in the microsporangium. 4 haploid microspores are produced from each diploid pollen mother cell by meiotic division. These microspores transform into pollen grains by developing into a characteristic shape and forming additional pollen grain walls. The wall of the microspore, known as callose wall, is broken down by an enzyme called callase, and the freed pollen grain later forms a resistant outer wall called exine and an inner wall called intine. Pollen grains have various shapes and sizes and surface markings that are characteristic of the species. The study of the morphology and structure of pollen grains is called palynology. The outer wall of the pollen grain protects the male gametophyte during their movement from the stamen to the pistil of the flower. The pollen grains move to the stigma of another flower or another plant. Since the cross-pollination is carried out by external abiotic, that is wind and water, or biotic, insects, birds, etc., agents, the pollen grains need a protective outer covering. The anthers break open to release the pollen grains once the pollen grain mature and are ready to be pollinated. Each pollen grain contains 2 cells, a non-reproductive vegetative cell that produces the pollen tube and the reproductive generative cell that forms two sperm cells. The female gametophyte or megametophyte, also called embryo sac, develops within the ovule enclosed by the ovary at the base of the carpel in a angiosperm flower. The megametophyte originates from the megaspore mother cell, which undergoes a meiotic division to produce 4 haploid megaspores. Out of these, 3 megaspores degenerate and the remaining one enlarges to produce the embryo sac. The haploid nucleus of the megaspore undergoes 3 mitotic divisions forming 8 nuclei within the enlarged embryo sac. In general, out of these 8 nuclei, 3 nuclei form 3 antipodal cells at the opposite end, 3 form 1 egg cell and 2 synergid cells at the top of the embryo sac, and 2 nuclei remain at the center of the embryo sac. This female gametophyte is now ready for fertilization. After pollination, the pollen grains land on the stigma of the ovary. Mature stigmas of many plants secrete sugary fluid on their surface. The moist stigma exudates stimulate the pollen grains to germinate and form the pollen tube. Fertilization is strictly regulated biochemically at this stage to ensure reproduction by the pollen grains of the flowers of the same species. Accordingly, there are compatible and incompatible crosses. The stigma promotes germination and growth of the pollen tube of only compatible pollen grains. Pollen germination and growth of pollen tube are aborted at various stages in an incompatible cross. For example, in pollination by wind, several types of pollen grains belonging to different species may be carried by the wind, and all of them may land on a stigma. If we take a cereal like maize, the stigma of maize may receive pollen grains of wheat or sorghum also, as all of them are carried by the wind. However, the initial biochemical recognition reactions on the stigma allow the germination of only maize pollens and abort others. Similarly, plants which are self-sterile to prevent self-pollination also inhibit the germination and growth of pollen tube. Out of the 2 cells in the pollen grain, during germination, the vegetative cell forms a tubular protrusion breaking through the walls of the pollen grain. This protrusion or pollen tube grows through the nutrient-rich style until it reaches the bottom of the ovary. The generative cell is carried by the pollen tube and it divides later to form 2 male gametes. The pollen tube may penetrate the ovary in one of the 3 different places depending on the type of the plant. On the basis of the place of entry of the pollen tube, 3 different types of fertilization are recognized. In porogamy, the pollen tube enters the ovary through the opening in the ovary called micropyle. This is the most common type of fertilization in angiosperms. In chalazogamy, the pollen tube enters the ovary through chalaza present at the base of the ovary. This type of fertilization is seen in plants like casuarina. In mesogamy, the pollen tube enters through the integuments at some point in the middle portion of the ovary, neither at the top nor at the base. Mesogamy is found in plants like cucurbit. The pollen tube may enter the embryo sac inside the ovary in one of the three ways. It may enter between the embryo sac and one synergid like in cardiospermum, or it may enter directly into a synergid like in oxalis, or it may enter between the egg cell and one synergid like in phagopyrum. Fertilization in angiosperms is unique since it results in fusion of two male gametes with two separate nuclei in the embryo sac. The fusion of one male gamete with the egg cell is syngamy that results in the formation of a zygote. Fusion of the second male gamete with the two polar nuclei is triple fusion, resulting in the formation of a triploid cell that develops into endosperm. The two polar nuclei fuse with each other to form a secondary nucleus before they fuse with the male gamete to form a triploid primary endosperm nucleus. Syngamy precedes triple fusion during fertilization. The fusion of the two haploid nuclei of the female egg cell and the male gamete restores the diploid number in the zygote nucleus. The zygote undergoes further mitotic divisions to form the embryo. The triploid cell formed by the fusion of the second male gamete develops into endosperm, which serves as the nutritive tissue. The other cells of the embryo sac, the antipodal cells and the synergids, disintegrate soon after fertilization. The petals and the sepals of the flower wither away after fertilization and the ovary develops into a fruit. The fruits may be fleshy and juicy like in mango, orange, papaya, etc., or the ovary wall may fuse with the seed like in cereals. The ovule develops into a seed inside the fruits. The seed develops a seed coat or testa from the integuments of the ovule. The seed enclosing the embryo and the endosperm also becomes dehydrated before it is dispersed from the parent plant. Parthenocarpy is the development of the fruit without fertilization of ovules. Parthenocarpy is either natural or artificially induced for commercial advantages. Parthenocarpic fruits are seedless and have only the fleshy portion of the fruit like in parthenocarpic papaya or watermelon. Seedless fruits are also naturally produced by another process known as stenospermocarpy, which is different from true parthenocarpy. In stenospermocarpy, normal pollination and fertilization of the flower occurs, but the development of the embryo is aborted at an early stage that leads to the production of a seedless fruit. Table grapes like Thomson grapes develop this type of seedless fruits by abortion of the embryo. Stenospermocarpy is also found in watermelon fruits. Parthenocarpy is either natural or artificially induced. Under natural conditions, parthenocarpy may occur due to mutation in some plants. In wild parsnip, 20% of the fruits produced are parthenocarpic. Natural parthenocarpy is advantageous to plants like parsnip since certain herbivores prefer seedless wild parsnip fruits, and this prevents seed predation that results in loss of seeds. Seed predation is a condition in which the seeds are damaged and made non-viable by some of the animals which feed on the seeds as exclusive source of food. Another plant, Utah juniper, also produces parthenocarpic fruits to prevent seed predation by birds. Two types of parthenocarpy are distinguished as stimulatory parthenocarpy and vegetative parthenocarpy. Stimulatory parthenocarpy requires some stimulation like pollination for parthenocarpy. If a plant does not require any stimulation for producing parthenocarpic fruit, then it is vegetative parthenocarpy. Seedless cucumbers are produced by vegetative parthenocarpy. Some triploid hybrid plants are genetically sterile and do not produce seeds, although they can develop fruits. In watermelon, crossing a diploid parent with a tetraploid parent produces a triploid plant. The seeds of the triploid plants are sterile, but they can produce seedless watermelon. Similarly, banana fruits are also produced by triploids. Since seedlessness is a preferable trait in fruits, many artificial methods like genetic modifications or hormonal treatments are used to produce parthenocarpic fruits. Seedless watermelon, papaya, tomato, grapes are some of the commercial fruit crops in which parthenocarpic fruits are produced by artificial methods. Replacement of natural sexual reproduction by asexual reproduction without fertilization is called apomixis. In the absence of sexual reproduction, there is no mixing of genes, and the offspring produced by apomixis are identical to the parent plant. Propagation of angiosperm plants like allium by bulbs or dahlia by corms are examples of apomixis. In flowering plants, apomixis is also referred to as agamospermy, that is, clonal reproduction through seeds without involving gametes. There are two mechanisms by which agamospermy occur. In gametophytic apomixis, the embryo arises from the unfertilized egg cell by parthenogenesis. Development of embryo without fertilization is called parthenogenesis. The embryo remains diploid when the egg cell does not complete meiosis. In some plants, the embryo may arise from a haploid egg cell and it is called haploid apogamy. The haploid embryo will produce a haploid plant, and this haploid plant will not propagate further. In sporophytic apomixis, an embryo is formed directly from nucellus or integument tissue of the ovary wall. This is also called as adventive embryony, and it is common in several species of citrus and mango. In vegetative apomixis, plants propagate by vegetative parts like bulbils, as seen in plants like allium and agave. Fertilization in higher plants ensures mixing of genes in the offspring through sexual reproduction by the fusion of male and female gametes. In angiosperms, the male gametes produce an endosperm nucleus along with the production of zygote. Fertilization initiates the production of fruits and seeds in plants. Production of embryos and fruits can also happen naturally without fertilization in some of the plants.